Thank you for joining on time for the sessions. This is Dr. Suresh here. I'm a consultant pathologist at the Dr. GVN Cancer Center, Tiruchirappalli. It's a pleasure to invite all of you to this workshop, A2Z of Cancer Genomics. Many of you may be already aware that this workshop is a part of a series of pre-conference events leading up to a two-day scientific conference, Focus 2025. Which is to be held at Courtyard Marriott on 31st May and 1st June. So this is a part of that pre-conference event. And this conference is focused on precision oncology topics like genomics, stereotactics, and robotics. So coming to our conference, you all know this is conducted as a hybrid format. So the initial part will be a series of 5 5-day sessions. All of which are online. And at the end of it, on the 31st of May, moderating today's session to kindly introduce today's speaker, Dr. Arun Kumar, please. Yes, sir, please take over.
Good evening, everyone. Dr. Arun Kumar is a scientist and in molecular genomics and has plenty of years of experience in molecular oncopathology. And he'll be presenting today's session on the basics of what is NGS, what is standard sequencing, and what are the basic terminologies that are used to ensure that everybody is comfortable with the language that is used. One request is to make sure everybody interact. You have also been categorized into small groups with a mentor. So it is essentially to encourage participation and to clarify any doubts. So wish you all the success and hope it will be a useful session. Dr. Arun, please. Thank you.
Thank you, Dr. Rajesh. Very good evening to all the participants. First of all, a very warm welcome to this course. I think we strongly believe you will benefit a lot from this course because we want to cover all the basics, and at the end of the course, we feel that you would be able to participate actively in a molecular tumor board, maybe even conduct your own molecular tumor boards. And you'll have a fair understanding of how precision oncology is practiced. So, quick thing, I'll just share my screen if, let me know if my screen is visible and I'm audible.
Very much.
Thank you so much. So, very good evening again. So, I'm Arun Kumar. I'm a molecular scientist. I have been trained in the molecular oncology. And I've held various posts in different firms, and currently I'm the medical head chapter lead for Roche Pharma in India. So I'm a molecular oncologist by training, and I'm actively involved in many tumor boards, and I've helped set up multiple molecular tumor boards across India. So in today's talk, I'd be covering 3 major topics or 3 sections. One, I'll give an introduction to genetics per se. We'll revisit basic concepts of genetics. And then we'll have a session on technologies, or specifically genomic technologies, which is where some everyday terminologies we'll try to understand in terms of technologies, so that it helps us in interpretation of the result, understand appreciate a report in a much better fashion. And finally, the third part will be on molecular oncology, which is the core of interpreting a genomic report, or which is the core of making therapeutic relevance from a genomic report. So without much ado, this is going to be a bit content heavy. I would suggest that in the interest of time, if you have any specific questions, you please drop in the chat box. If something is super complicated and you want me to repeat it then and there, then please raise your hand. If not, at the end of every section, we can have a quick 5 minutes Q&A before we move forward. So I hope that's fine with everyone. But if you're confused, or if something is not clear at all and you want me to repeat it then and there, please don't hesitate. Just raise your hand and ask me to repeat it. All right. So These are my disclaimers. I'm presenting this only for academic purposes, and I own full responsibility of what is being presented here, and my employer does not have any role or influence in the contents of this presentation. So we'll start with the very basics. I spend a minute in most of my talks on this because understanding, well, this talk is this slide is on genomic organization. Understanding the levels of genomic organization is very important for us to appreciate what type of technologies to be used, appreciate the complexities behind genetic aberrations. So at the base, we know DNA, the genome is composed of a polymer, biopolymer called DNA, deoxyribonucleic acid, which is essentially your sequence of 4 monomers called adenine, guanine, thymine, and cytosines, which are the nucleotides. Now, each human being has 3 billion bases in one strand of the genome. Then this long stretch of DNA molecule is wound around certain protein particles, and those protein particles are called histones. And this combination of histone and a DNA is called as a nucleosome. This winding around the histones helps in the compacting the genome into a cell, into the nucleus of a cell. And this nucleosome is further condensed into a 300 and a 700 nanometer particle, and the most condensed form of the genome is called as the chromosome, which is the classical X-shaped structure. Now, this chromosomal structure of the genome exists only when the cell is dividing. Otherwise, in a normal healthy interface cell, when the cell is actively doing its life of metabolism and so on, it is generally there somewhere between this 300 and 700 nanometer particle. And as and when a gene has to be expressed, it becomes a double-stranded DNA. The uncoiling happens and the double-stranded DNA is formed. Now, the point of this slide is that you can have genetic aberrations, or in technical terms, it's called the mutation, could happen at a DNA level, happen at a nucleosome level, happen at a 300 or a 700 nanometer level. It can also happen at a chromosomal level. Now, the type of mutation or the scale of the genetic aberration is important for us to know so that the appropriate genomic technology can be applied to identify or detect those genetic aberrations. All right. Now, this is a slide I would request all of you to have a mental picture of this, because I'm going to keep referring to this slide in various parts of my talk so that you would be able to better appreciate. Right. Now, there are coding and non-coding region in a genome. So first of all, what is a genome? Genome is the complete set of the DNA that is there in a particular cell. Now, that complete set of DNA is composed of coding and non-coding parts. The coding parts are called the genome, genes, and the non-coding parts are the non-coding intergenic spaces, right? So when you have a chromosome, it is made of genes and intergenic spaces, right? And a gene is having a specific composition in eukaryotes, we being the eukaryotes. Okay, so the gene is the coding part of the genome, which is composed of exons, which are the coding part of the gene, and introns, which are the non-coding part of a gene. So it is important for us to understand the difference between an intergenic space and an intron. Intron is there within a gene, whereas an intergenic space are spaces between genes in a chromosome, right? And these introns are removed away when the gene is getting expressed. In addition to this, you have the transcriptional control region, you have the promoter region, which basically tells that, hey, look, this is where the gene is starting, and from here is where the RNA synthesis should get started. And it also has a sequence which says, okay, here is where you have to stop the RNA sequencing or the RNA synthesis, and that is called as a transcription termination region, right? Now, moving forward, the way the gene is expressed is like this. You have, I just mentioned how the structure of a gene, and this gene, by the process of transcription, produces a mRNA. Now, here, I would like to give you a definition. I said I defined what a genome is. Genome is the entire DNA material of a particular cell. Whereas a gene is that part of the genome that codes for a functional RNA, right? Early on, we used to study gene codes for a functional protein, but with the advancements in transcriptomics, we understand that there are many RNA which have a biological function which need not actually be done through a protein. So those RNA that produces a protein is called as an mRNA, but you do have small RNAs like microRNAs, snRNAs, etc., which have biological functions, but they do not produce a protein. So those RNAs are also coded from a gene. So gene is a segment of the genome that codes for a functional RNA by the mechanism of transcription. Now, this RNA is processed, and from this RNA, through the process of translation, a protein is produced, right? So it is the information or the sequence of the DNA that determines the sequence of the RNA, which determines the sequence in the amino acid sequence in the protein. All right. Now, only a subset of the gene is relevant in cancer. Only a subset of the number of genes that we have in the human genome is relevant in cancer. So the total human genome has somewhere around 22 to 25,000 genes. And you see, it's only that 1.5% of the total genome that is composed of genes. The remaining 98.5% of the genome is your intergenic spaces. You remember in the second slide that I showed you, you have a chromosome which is composed of genes and intergenic spaces. It is actually more intergenic spaces and very less number of genes. There are multiple hypotheses of the function of these intergenic spaces. I'm not going into the details of it. And this 1.5% of the human genome codes for a functional RNA, which is composed of close to 22 to 25,000 genes. And among these genes, only a few hundreds are involved in directly or indirectly in cancer, right? Having seen what the organization of the genome is and the structure of a gene and how the gene gets transcribed, let us now go on to see what are the classes of genetic aberrations that one can see. So genetic aberration is defined as basically called a mutation, which is a change in the sequence of a gene that results in an altered function of a protein. For example, there are four major classes of genetic alteration, particularly which can drive tumor growth. The first and the most common type of alteration is called as a base substitution. It is also called as a single nucleotide variant, SNV, right? Single nucleotide variant or a base substitution. Now, what happens here is in the sequence of DNA, one nucleotide becomes another. So in this case, A has become a C, which leads to a change in the amino acid sequence of the protein. For example, your BRAF V600E is an example of a single nucleotide variant where one amino acid becomes another. So it's just a single nucleotide that changes from one nucleotide to another at the DNA level. Almost 98% of all known human genetic variants belong to this class of SNVs. Second, you have what is called as insertions and deletions. Now, insertion, as the name implies, say one or a few bases can get inserted or a few bases can get deleted. Best examples are your EGFR exon 19 deletion, where somewhere around 40 to 50 nucleotides get deleted in the EGFR gene. You can also have insertions and deletions at a large chromosomal level. Remember the 300 to 700 nanometer particle level, where exon level insertions and deletions can also happen. So insertion deletions can be at a nucleotide level. It can also be at an exonic level. Just remember that. We'll talk a bit about that when we come to the technologies. You can also have copy number alterations. Now, ideally, every gene we have two copies, one from the father, one from the mother, right? But in certain conditions, the copy number increases. For example, you would have heard of this HER2, which is a gene involved in the growth of a cell. And the HER2 gene, instead of two copies, can become 30, 40 copies in the chromosome or in the genome of that patient. that leads to excessive activation of the HER2 pathway. So this is an example of a copy number alteration, or it's also called as a copy number variant, CNV. These are all synonymous terms. Just have in mind SNVs is single nucleotide variants, whereas CNVs are copy number variants. It's also called copy number alteration. And lastly, please have a watch here. It's called as a gene rearrangement. Now here I'm going to define two very important terms. One is a rearrangement and the other is called as a fusion. Rearrangement, or it is also called translocation, is a physical event where one chunk of DNA from a chromosome gets cut and gets pasted in a different part of another chromosome or the same chromosome, right? It's a cut and paste mechanism. One chunk of DNA gets cut from here and gets pasted elsewhere. Now, if that chunk of DNA is a gene, it's getting cut from chromosome A and gets pasted in chromosome B. But the location where it is getting pasted is in the proximity of another gene, let us say gene B. Then it produces a chimera protein. That is a protein that is composed partly of gene A and partly of gene B. Now, this process of cutting and pasting is called rearrangement or translocation. The process of those translocations that lead to a chimera protein being produced is called as a gene fusion. So what do we understand here? Not all rearrangements need to necessarily lead to gene fusion, but all gene fusions are a result of some form of a physical rearrangement or a translocation that has happened, right? I hope this concept is clear. So examples include your ALK fusions, EML4 ALK fusions, BCR-ABL translocation fusions. You have NTRK fusions, etc. Now, as we do more and more genomic profiling, we see that there are also genes where fusion is not produced, but a rearrangement alone happens, right? So I think in one of the case studies I have included that. Let us try to understand. So 4 major classes of genetic aberrations: single nucleotide variants or base substitutions, insertion deletions, copy number alterations, and gene rearrangements. Now, the single nucleotide variant, which is the most common type of a variant, can be there in any part of the gene. It could be there in the promoter region, it could be there in the exons, introns, 5' untranslated region, and it could also be there in the 3' untranslated region, which is the 2 sides of the gene, right? Transcription initiation and the termination slide. Now, promoter region is the region where RNA transcription starts. So any mutation in the promoter region can directly affect the way in which the gene is expressed. For example, your TERT promoter mutation in glioblastoma or CDH gene mutation, promoter mutation in prostate cancer leads to differential expression of these genes in that tumor, leading to oncogenesis, right? There could also be methylation or epigenetic changes that can happen in the promoter where differential expression of the gene can happen. You could have single nucleotide variants in the exons. I think one of the examples we just saw was that BRAF V600E or the EGFR T790M, because these mutations or variants happen in the coding region of the gene, which is the exon. You could have variants in the introns also, which can affect the process of splicing. Now, splicing is the process where the introns are removed from the mRNA and the exons are joined. Now, for that splicing to happen, there are specific bases in the exons and the introns which specific splicing proteins recognize. If there is a variant there, that leads to improper splicing. Can I ask any one of the participants here to give me an example, a very common example of such variant that is seen in lung cancer? Can anyone quickly tell me, do you know any such intronic mutation that causes pathogenesis in non-small cell lung cancer? Anyone?
Skipping mutation, sir?
Absolutely right. So MET exon 14 skipping mutation is one such example where the mutations happen in the exon-intron boundary, right? So if you remember the gene diagram that I showed you, exon followed by intron. Now, between the exon and intron, that region is called the boundary. Certain variants in that intron region, in that boundary, which could also be there in the intron, prevents that intron 14 to be spliced out. So that is why it's called exon 14 skipping mutations. It skips the exon 14 and the intron 14 to be spliced out, right? So that is one great example. There is another polymorphism in the FGFR2 gene, which is also involved in the pathogenesis in certain types of tumors. Then you can have variants in the 5-UTR, for example, this plus 2756 C to T mutation in the c-MYC gene, or in the 3-UTR where there is a polymorphism in the interleukin 23 receptor gene that can affect the mRNA expression or microRNA binding, etc. So essentially, genetic variants can happen in any part of the gene, and many of these variants can cause, since we are related to oncology, many oncological phenotypes. Now, next definition here, I'm sure most of you know it, but for the sake of genetic variants can be germline or somatic in origin, right? A germline variant is the variant which is inherited from the parents, right? So the mutation is there in the father or the mother, and that gets propagated to the next generation. So germline variants are such variants, which means that all the cells of the body of that individual has the variant or that mutation. Whereas somatic variants are those variants which are acquired in nature during the course of the lifespan of the individual. For example, your EGFR exon 19 deletions, or BRAF V600E, or the KRAS G12C, or your PIK3CA mutations, all of these are somatic variants which are acquired in nature and specific to that tissue where the tumor is there. So, to do a germline testing, you can take any cell of the body, the easiest one being your WBC or your buccal swab. For a somatic testing, you need to do the testing only in that particular tissue. You cannot use any other tissue, right? Right. So let us spend a bit of time in the germline mutations that you see in cancer. Many of this would be definitely discussed in the talk later, which is there on familial cancers and genetic cancer syndromes, but I'll just give a primer here. Germline mutations account to almost 10 to 15% of all known cancers. In other words, 10 to 15% of cancers are inherited in nature. There are wonderful studies both globally and many from India, great studies from India that have shown the frequency of such mutations in the Indian population. Why do you need to understand this? Because if you do a germline testing, it helps in prognostication. Some of these mutations help in therapy decision making, screening and management, and it also helps in cascade testing, that is testing other family members of the particular patient and help us devise screening and preventive strategies. So just to give you an idea about germline, there are different types of inheritance mechanisms. Commonly the autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and the mitochondrial type of inheritance, particularly for single gene disorders, right? Especially in cancers, the autosomal dominant and autosomal recessive are very important. So I'll quickly show what that is. So autosomal dominant are those genes where mutation in any one copy of the gene is sufficient to cause the phenotype. For example, in this case, if the mother father has 2 copies of the gene, the mother has 2 copies of the gene. If one of the copy of the mother's gene is mutated, the mother has the phenotype, and those children who inherit that mutated copy of the gene also have the phenotype. So those children who have this have one healthy copy and one affected copy. So in other words, autosomal dominant genes are those genes where a single copy is sufficient to cause the phenotype. On the other hand, autosomal recessive genes are those genes where you need to have two copies of the mutated gene to cause the phenotype. So, if this can happen only when both the parents are carriers. If the father is a carrier, mother is a carrier, there is a 25% possibility of one of the child being the carrier. So, if the child one of the child being affected, because that child inherits both the defective copies of the gene, there are 50% probability that a child becomes a carrier because he or she inherits one copy of the mutated gene and a healthy copy of the other gene. So, carriers do not exhibit the phenotype, whereas those who get both the copies of the mutated gene show the phenotype or the disease. So, most of the cancer syndromes are autosomal dominant, very rare to see an autosomal recessive trait in a cancer syndrome, but most of the cancer syndromes are autosomal dominant in nature. These are some features suggestive of hereditary cancer syndrome in a case: early onset, close blood relatives with cancer, multiple primary sites in the same organ, multiple tumor types or multiple primaries in the same person, occurrence of certain types of adult cancers, benign growth, skeletal abnormalities, autosomal dominant inheritance, or certain features suggestive of hereditary cancer syndrome. Now, next I would like to introduce another very important terminology called penetrance of a gene. Okay, so what does this say? Let us say a person has inherited a defective copy of an autosomal dominant gene, let us say BRCA1 mutation. Now, what is the probability that or likelihood that the person develops a malignancy in his or her lifetime upon inheritance of this mutation is the penetrance of the gene. Now, not all the genes have the same penetrance. So, we classify genes as high, medium and low penetrance. So, high penetrance genes include your BRCA1, BRCA2, PALB2, C, TP53, etc. Moderate penetrance genes like ATM, CHEK2, NF1, and low penetrance genes typically your MLH1, MSH2 kind of genes. So, what do I mean by this? I'll show it to you in numbers. The lifetime risk of a person having a BRCA1 mutation to inherit a malignancy, to develop a malignancy is 71%. For a BRCA2 mutation, it is 69%. On the other end of a spectrum, a person with a mutation in the MLH1 or MSH2 gene, the lifetime risk of developing a malignancy is less than 15%. So, this is a low penetrance gene, whereas BRCA1, BRCA2 are high penetrance gene, whereas CHEK2 or RAD51D are median penetrance genes. So, I hope you understand this concept of penetrance. So, this helps a lot while giving counseling for patients, especially when you detect a medium or a low penetrance gene in a family member. It helps in genetic counseling. I'm just listing out, I'm not going to go to details here. These are some common hereditary cancer syndromes, the organs they affect and the type of the genes that cause this syndrome. Hereditary breast and ovarian cancer syndrome caused by BRCA1, BRCA2, or a Peutz-Jeghers syndrome by STK11, Li-Fraumeni by TP53, Lynch, etc. I'm sure these will be covered in detail in the other talk on familial cancer syndromes. Next, I will visit the somatic mutations in cancer. So, first we saw the germline mutations. Now, we'll see the somatic mutations. So, somatic mutations, as we saw, are acquired mutations, and they could also be epigenetic modification that changes the expression of a gene because of changes in the promoter region or the histones of the particular histones particle where the gene is there. Somatic mutations can also be point mutations, your SNVs, gene fusions, amplifications, deletions, post-translational modifications, or epigenetic modifications, typically your histone modifications and DNA methylations. So, how does somatic mutation cause cancer? As we know, DNA to RNA to protein is what is the flow of genetic information. And what we need to understand is these proteins, multiple proteins come together or in an orchestrated fashion and produce and are involved in signal transduction pathways, right? This is the last part of my talk, which I will talk in detail. You may have heard of the PIK3 pathway or the RAF pathway, which are involved in proliferation, survival of the cell. Now, if at all there is a mutation in the DNA, say your EGFR exon 19 deletion, that alters the DNA, alters the RNA, and alters the protein, leading to alteration in the pathway or the control of the pathway, leading to uncontrolled proliferation and survival. So, what does this mean? To understand why why the pathway is affected or what pathway is affected, we need to understand what type of mutation has happened in the genes that are coding for the proteins involved in this pathway, right? So the types of mutations is what we saw. Those genes which are involved in these pathways, specifically involved in the pathways related to tumor, can be classified into two as oncogenes and tumor suppressor genes. Please pay attention here. It's a super important definition. So proto-oncogenes are genes involved in cellular pathways. Particularly, these genes are involved in activation of cellular pathways, right? So your EGFRs, your ALK, your FGFR, your HER2 are all proto-oncogenes, which when become mutated becomes an oncogene, and mutations in these genes are mostly gain of function mutation, right? Only when you have a gain of function mutation can the protein get super activated. That is, it acts more than what it should normally perform and leads to cellular pathway activation, right? So as a thumb rule, for oncogenesis, proto-oncogenes undergo gain of function mutation. In contrast to this, you have the other set of genes called tumor suppressor genes. These genes are involved in blocking cellular pathways and cell cycle, right? So these genes typically prevent a malignancy happening, which means loss of function of these proteins coded by these genes lead to excessive activation of the cellular pathway or cell cycle, leading to tumorigenesis. So mutations in these tumor suppressor genes are loss of function mutations. So two things: proto-oncogenes have gain of function mutations, tumor suppressor genes have loss of function mutations. I hope this was clear. So some clinically important proto-oncogenes are shown here. Growth factor receptors, for example, your HER2, EGFR, MET, RET, FGFR, growth factors. So these are receptors, and these are the growth factors which bind to the receptors, like fibroblast growth factor, FGF2, FGF3, CIS, etc. Signal transduction factor with kinase activity like ABL, RAS, CDK4, anti-apoptic factors like BCL2 or MDM2, transcriptional factors like MYC, FOS, JUN, etc. These are all some of the proto-oncogenes which get mutated to become an oncogene. And these are some of important tumor suppressor genes like TP53, RB1, your CDK, BRCA1, your MLH1, APCs, etc. So these are all some of the clinically important tumor suppressor genes. So loss of function mutations happen in these tumor suppressor genes, whereas gain of functions mutations happen in this proto-oncogenes, right? So now we will quickly move on to breakout rooms, but maybe before we move there, are there any questions from the audience? Do you want any specific part to be repeated, something like that? Or is it super clear?
I think you kept it very simple and eloquent. And just to provoke some questions, you had mentioned about this copy number variations and insertions, deletions, and stuff like that. And we even emphasized on small insertion deletion and large insertion deletions. And the session is going to be more and more on NGS, I understand. So, can you mention some of the limitations of NGS while testing for these different types of mutations?
Absolutely, sir. I mean, this is going to be a big chunk of my next talk, in fact. So I'll be comparing fish, karyotyping, IHCs with NGS. So if you don't mind, I will explain that because it's part of the uh, my talk only in the next one.
Arun KK here from
Hi,
Madurai.
doctor. Hi, sir.
Hi, Arun. Nice talk.
Thank you.
Just you were talking about the mutations.
Yeah.
So we know that each gene has two loci on both chromosomes.
Yes, doctor.
So just for the benefit of the postgraduates, I just want you to say two things. One, is it necessary that the loss of function has to occur on both chromosomes, the whatever we call the double hit hypothesis? And if that's the only case that can defunctionalize a gene, then what is this concept of monoallelic and biallelic mutations for a particular gene?
Good. I think it's a very important question. Okay, that gets a bit complex. I hope the our participants are attentive here.
You probably can repeat you are
Yeah,
probably your way you explain your language is much more student friendly. So I
sure,
think I have just gone a bit overboard. You can decode my question to the PG level and again please clarify it for us.
sure. So I'll explain that. Now, what Dr. KK is trying to ask is, we have two copies of the gene. Is mutation in one copy alone sufficient to cause the pathogenesis? In simple terms, that's what he's saying. Now, say for example, BRCA. We said BRCA gene is autosomal dominant, which means you inherit the patient inherits one copy of the gene, mutated gene. Is that sufficient to cause cancer? The answer is, for certain genes, it is required to get a double hit, right? So what it means is that when one of the BRCA gene is mutated, the other healthy gene compensates for its activity as much as it can. Okay, I'm now only talking about BRCA. As in biology, there are exceptions in every rule when I say it's a rule, right? So let us just talk about BRCA, which is the most common one. When one gene is defunct, it produces a protein which is not functional at all. It just gets degraded. The other healthy gene compensates for this until the day the patient develops tumor. Why does the patient develop tumor? Because where is BRCA involved? It's involved in DNA replication. It makes the other healthy gene also get defected. It induces mutations, it induces deletions in the healthy gene, which is the second hit required for tumorigenesis. That is why you would see that some patients develop, who have a BRCA mutation, develop breast cancer at the age of 30, whereas other patients develop it at the age of 45 or 50. Why? Because the time it requires for the second hit varies with every individual, right? So this is the dual hit hypothesis, where first hit a person might inherit, the second hit happens over a period of time when the healthy copy also gets affected. Does that answer, Dr. KK?
Yeah, but so then what is this concept of monoallelic and biallelic loss of function?
Okay, this is a bit complex again. Monoallelic and biallelic loss of function is to do with clonality of the cell. Particularly if I can take leukemia as an example, there is a gene called, if I'm right, NPM1, wherein one healthy copy alone, one mutated copy, both are somatic mutations, they are not inherited. Now within a cell, NPM1 gets mutated, leads to tumorigenesis. Now that has a different prognosis. In one subclone of the cell, the second healthy copy also gets a second mutation. That becomes a second hit or a biallelic loss. So both the copies of NPM1 are lost in that clone of cells, and that has a much poorer prognosis. Now, this single and double hit of this specific set of genes like NPM1 or few others, even in EGFR this concept is coming up, is required for us to take clinical decisions like whether to go for a bone marrow transplant, whether to go for a specific high-intensity chemotherapy, etc. So understanding whether single gene or both the genes are hit is required in specific clinical scenarios like ALLs or certain EGFR mutated tumors.
Thank you, Arun. Thank you so much.
Yeah.
So
Somebody,
there was a question on penetrance and where to find the details of penetrance. It's in the literature. I think you can explain that.
absolutely. It's in the literature. There is no one place. You can just search for lifetime risk of the gene name. You will get the penetrance of it, sir.
Just to clarify, that double hit hypothesis or Matson's hypothesis, what it is called as, is in the context of loss of function in tumor suppressor genes.
Yeah.
And though it is a simplistic viewpoint, there are, of course, as in any other thing, when we go into the detail, for individual genes, there are certain differences. It's a very complex area. I think you have tried to explain it. We can go into those individual chat rooms.
Please, yeah. Very quick 5 minutes. Let us not spend a lot of time there. Very quick 5 minutes. Organizers, can you just switch on the break rooms?
Arun sir, Arun sir, this is Dr. Kumar from Salem.
Oh, hi, doctor.
Hi, sir. So you were just mentioning about 4 classes of genomic alterations.
Yes, doctor.
So just can you be elaborate on the 4th rearrangement on gene fusions?
Sure. You want me to show? I think I don't
I think
have it.
the slide number 7.
Yeah, but I'll just explain. You remember the chromosome, right? Chromosome has genes. Now, one gene gets cut from chromosome 1 and gets pasted, let us say, in chromosome 9. Yeah. This physical movement of this gene from chromosome 1 to 9 is called translocation or rearrangement. Translocation is equal to rearrangement, right? Yeah. If this happens, if this gene A from chromosome 1 getting pasted in chromosome 9 happens between near a gene B. Okay, gene B is here. Unfortunately, gene A gets pasted here. Just as the gene B ends, gene A gets pasted there. Okay. This will produce a fusion protein, which is your BCR, ABL, EML4, ALK. Right? So these are fusion proteins. So not all translocations or rearrangements lead to fusions, but some of them lead to fusions, and that can be pathogenic or oncogenic in nature. Is this clear? So
yeah, crystal clear, sir. Thank you.
Rearrangement is a physical movement. Fusion is the effect of the rearrangement. Exactly. So that's what it is.
Thank you, sir.
Thank you. I hope you all were able to have a good discussion. So there were a few questions. Can we just quickly, any one group, whom shall I select? Any some number. Okay, let us say group 5. Can you kindly answer these questions? What did you discuss? Any questions, any answers you have? Anyone from group 5? Hello? Sorry, am I audible?
Yes, sir, you're audible,
Yes,
sir.
doctor. Yes, doctor.
Okay.
5, any one of you all?
Bhagat Singh, you
Yes.
have been answering.
Yes, Dr. Subiksha.
Yes, sir. The Sanger testing is it is done, sir. So to determine the order of the nucleotides of the DNA sequencing. So, hello.
Yeah. I can hear, but order of the DNA sequencing was also seen in the NGS, right?
Yes, sir.
So why was Sanger done specifically?
Look for particular genetic mutation because they're looking at a germline, suspicious germline. You want to confirm if that is seen in that specific sequence.
Perfect. So because the first test, NGS, was a somatic testing.
Okay.
And in ALL, right, the somatic tissue is the WBC. So the first test was done in the blood sample of the patient. Now, what was the sample used for Sanger testing here to test germline? It was buccal swab. Why was it so?
For germline, because the
Yes.
VAF was more than 50%.
Correct. The reason we chose buccal swab and not a blood is because blood already has the somatic mutation. You won't be able to differentiate between somatic and germline if you again took blood. Whereas if you detected the same TP53 in a breast cancer tissue and you wanted to check if it's a germline, you can do a Sanger in the blood. But in this case, WBC itself is the somatic tissue. So we took a different tissue of the body, which is the easy one to pick was the buccal swab. So that if it is present there, because germline mutations are there in all the cells of the body, including your somatic cells. So to differentiate that, we need to take a different tissue to see if it was positive. Now, the last question, how do you explain the cascade testing? You saw the result of the cascade testing. Proband was positive, father, mother, and the brother were negative. How do you explain this? Anyone wants to take a stab at it? Any wild guess?
It's heterozygous.
It is heterozygous, no doubt. But if it is a germline mutation, either the father or the mother has to be positive, right?
Penetrance is less.
Okay, penetrance is the lifetime risk of developing the mutation. So, okay, even if the penetrance is less, the father or the mother should be positive, right? They may not have developed the mutation, developed the phenotype, in this case, some malignancy, but how can they be negative? So, in other words, is there a scenario where parents can be negative and the child can be positive for a mutation?
Very early mutation in zygote formation, is it
The
possible?
mosaicism?
De novo germline.
De novo, exactly. So the answer is exactly de novo or mosaicism. So that's a very interesting case. It's not so uncommon. I think somebody else, Dr. Shivkumar also was saying he just now had a case of a de novo mutation like this. So de novo mutations are those novel germline or mosaic genetic changes contributing to an increased risk of developing cancer. How does that happen? Generally, the parents are carriers or they have the mutation. But if the mutation happens during the spermatogenesis or the oogenesis, then the parent's body does not have the mutation. It's only that sperm or that ovum that has the mutation which the child inherits. If it is a mutation in the germ cell, all the cells of the child will have the mutation because the zygote in the single cell stage itself has the mutation. It could also be that the zygote develops a mutation, say at the 16 cell stage or 32 cell stage or even much later in the development cycle of the zygote. If that develops a mutation, then one lineage or one sect of the cells in the adult body has the mutation, which is what is called as a mosaic picture, right? You see mosaicism in certain inherited PIK3CA mutations. You see mosaicism in even Down syndrome, where one lineage of cells develop the mutations. The mutations was not there either in the sperm or in the ovum or in the parents, right? So these types of mutations are called de novo mutations, which is the mechanism by which a child can inherit or get a mutation which is not there in the parents. So many TP53 leaf hominids tend to be such de novo mutations, right? Cool. So I think we had a good discussion. Thanks for that. And let us move on to the second section of my talk, which is a bit technology oriented. In the first section, we saw the organization of the genome. We saw the structure of a gene. We saw how the information is passed. We saw various mechanisms of inheritance. We also differentiated germline from somatic and the types of somatic mutations that are there. And we also saw an interesting case where instead of being a germline or a somatic, it was a de novo mutation, which is the third form of inheritance, although rare, is still possible. Now, in this section, we're going to see how to detect these mutations, right? So, again, refreshing your memory to the genomic organization, remember the DNA, it gets wound into nucleosome as a histone plus DNA, which becomes a 300 and 700 nanometer particle, and you have the chromosome. Your gene, say 10,000, 20,000 bases long, is somewhere around in this 300 nanometer particle size, right? So that this is the size of a gene, and whereas chromosome is the most condensed form of the gene. Now let us see what are the techniques available to see genetic aberrations in these hierarchical forms of the genome. So you have point mutations or single nucleotide variants. You have large deletions and duplications, right? So again, you see here, SNVs is a single nucleotide variant. Small insertions and deletions we have seen. Large deletions is what I said, it happens at the exon level, which is at the 300 nano. So these point mutations are in this level of the genomic organization, whereas those large deletions and duplications of exons is at this 300 nanometer particle level. Now, NGS is great tool to detect mutations which are point mutations in nature. Whereas when you have large deletions and duplications, you need to go for other technologies like MLPA or fragment analysis, right? We'll talk in a bit about it. You also have translocations or gene rearrangements, and you have technologies like karyotyping, FISH, RNA sequencing of NGS used for detecting these translocations. Now, I'll visit a bit about karyotyping, FISH, Sanger, and NGS, and I'll also touch a bit about the sensitivity or what to do with RT-PCR. So, karyotyping is the genesis of, I think, the field of genetics, where you physically look into a microscope and see and count the number of chromosomes a particular cell has. I'm not going into the details. You're going to culture the cells. The cells could be blood, amniotic fluid, tissues, the somatic tissue, etc. You culture them, arrest them in the metaphase. Why? Because only in the metaphase of the cell division, the chromosomes are maximally condensed, and you would be able to appreciate the chromosomes, stain them, and do the manual counting in the microscope. This is the karyotyping, still the gold standard for many genetic disorders. Advancement of karyotyping, which is also a cytogenetic technique, is called as a FISH or fluorescent in situ hybridization, where you prepare large probes. Please note the word large. These are 100,000 bases long. 100,000 is what? A lakh or lakh and a half bases long, which encompass the entire gene. Large gene-sized probes, which are fluorescently tagged to a chemical molecule. So these probes, when you add it to a interface cell, for a FISH, you don't need to culture the cell, you don't need to make the cell division. In the normal interface cell, wherever the particular gene is, this probe goes and binds and gives a signal. Now, if the signals are separated, that means there is no gene fusion that has happened. If the signal are joined together, you see that a gene fusion has happened. I've shown an example of BCR ABL, the green being BCR and red being ABL. One copy of the cells BCR and the ABL are not fused, whereas the other copy of the cells BCR and ABL are fused together, right? It's a 922 translocation, right? So this is called as a FISH, much easier to do, highly focused, but unlike karyotyping, which you can scan all the chromosomes, here you look for very specific translocation or chromosomal breakage in question. So PML, RARA, BCR, ABLs, things like that, inversion 16, you can pick in this. Next comes the gene sequencing technologies, where you have two major classes of gene sequencing technologies. The classical Sanger sequencing, the other is the next generation sequencing. Sanger sequencing is a sequencing technique wherein you sequence every single nucleotide of the gene of a specific part of the gene. So in Sanger sequencing, it is still the gold standard because the accuracy is very high, but you can sequence only 500 to 700 bases at a given point of time. In one single experiment, you can sequence only 500 to 700 or max to max 1000 bases long, whereas your gene will be somewhere anywhere from 50,000 to 1 lakh or even 5 lakh bases long. So you can sequence only a very small segment of the gene in a Sanger sequencing. On the other hand, you have next generation sequencing, which is a vast improvement, but it may not be as accurate as Sanger sequencing, right? But that small 0.1% or 0.01% loss of accuracy can be compensated for the large section of the genome that is being sequenced. So you can sequence even the entire human genome of an individual in a single experiment. So if you are doing a targeted sequencing, say 100 genes or so, you can sequence 100 genes of 10 or 20 people in a single experiment. Whereas in a Sanger sequencing, you can sequence only a very small region of the gene, let us say 1000 bases or so, in one individual in a given experiment. It is this reason why human genome sequencing took almost 7 years to complete, right? With close to collaboration between 13 different laboratories, with more than 5 lakh or 6 lakh sequencing experiments being done in each lab to get the human genome first draft way back in 2001. Today, the same human genome sequencing we can do using next generation sequencing in a matter of 4 or 5 days time, right? So this is the major difference between Sanger and next generation sequencing. Now, there are many different chemistries of next generation sequencing technology, but I'll just give you an overview of what NGS technology is. So in NGS technology, you take a sample, say the blood or the tissue in terms of in case of a somatic testing. You chop the DNA, you extract the DNA and chop the DNA into short fragments. Depending on the type of sequencer, if it's an Illumina sequencer, you chop it to 150 bases long. If it is some other type of sequencer, you chop it to 200, 300 bases long. Basically, you chop those into fragments, the entire DNA. Then these are random fragments, remember. At this point of time, you don't know which fragment came from which gene. And remember, this is not from a single cell, right? When you extract DNA, you're going to extract from multiple cells. So you have multiple cells and multiple different fragments from each cell. Then you do what is called as a hybridization, where you develop, say, 50, 60 base long probes. Remember, fish had 1 lakh base long probes, whereas these are just 20 to 50 base long probes, which are very specific to that specific region of the gene. So each gene will be made of hundreds or thousands of probes, and those probes will be hybridized to these random fragments. Probes are also DNA sequences, or mostly in an NGS experiment, RNA sequences, which are hybridized to the DNA fragments, and they are pulled down. Where these probes typically have a small ion or a magnetic particle attached to it, where when you keep the magnet to the test tube, all the DNA and the probe complex come to one side and you wash away the remaining unbound part of the genome. Which means what? Suppose I'm interested to sequence only 100 genes. I design probes for those 100 genes and I wash away the remaining part of the genome, which is like 99.99% of the genome is washed away, and I pull down only those 100 genes, which I ligate with what is called as adapters and take it up for sequencing. This entire process can easily be actually automated. When I started NGS, we had to do everything by hand. Now you have beautiful robotic technology where you can do everything automated. But the analysis, the data that you generate out of NGS is what that requires real analytical skill set. What is the data that you get? You get millions of reads. Reads are these short fragments that were sequenced from a single experiment, and you have to make biological sense out of it. So it is that, and it is that part of the NGS which makes the experiment really difficult. Otherwise, most of these are automated. Now, with this background, I want to take you through two very important definitions or statistical parameters in an NGS experiment. It is called as the read depth and gene coverage, right? Now, there is a process called mapping where each read, so read is that short sequence that we get from an NGS based on the random fragment that we created. They are bioinformatically mapped to the human genome, right? And that you have a lot of statistical bioinformatic mechanisms or algorithms around it. Once you have mapped, you then look for the quality. And one such quality is called as the read depth. Read depth tells you how many times each nucleotide is sequenced or is represented in various fragments. For example, this is a gene and this is one nucleotide of the gene. This nucleotide is represented in 1 to 7 fragments, 7 different random fragments that were generated and sequenced. So the depth of sequencing here is 7x, right? Whereas the depth of sequencing for this nucleotide is just 2x. The depth of sequencing for this nucleotide is 1x. And the depth of sequencing for this nucleotide is 0x. So read depth tells you, in other words, how many times this nucleotide is sequenced, right? So this is a very important parameter. As a thumb rule, for a germline mutation, we aim for a depth of around 250x, which means that every single fragment, every single nucleotide will be sequenced or be represented in at least 250 such fragments. Whereas for a somatic mutation, the read depth we aim for is 500x and above. So the median should be a 500x depth, right? So minimum of 500 fragments need to represent the mutation. Is this concept clear? Read depth is the number of times a particular nucleotide is sequenced in that particular NGS experiment. Now, gene coverage is, so you remember, we captured the genes using the probes that we had designed. Now, these probes must be for the entirety of the gene. But sometimes you do have certain complexities like pseudogenes, which makes the probes to be designed very difficult. So sometimes some part of the gene will not be captured adequately, and those are not covered. So let us say if this gene is of length 1000 bases, 900 bases are sequenced, whereas 100 bases are not sequenced. So the coverage of this gene in this experiment is 90x, 90%, whereas 10% of the gene is not covered. I hope this concept is clear, right? It can also lead to another, I'll take one more complexity here, show one more complexity here. Sometimes certain part of the genes may not be covered. You can have a 0x coverage, 0% coverage, even when you have designed the probes adequately, right? That can happen when you have a gene deletion. When the gene is deleted or you have an exon level deletion, you may get a 0x coverage there, in that 0% coverage there. So a bioinformatician who is analyzing the NGS data should be able to differentiate the non-coverage part of the gene from a biological deletion from an error in the NGS or error in the probe design of the NGS, which is the reason why we say NGS is not very effective or highly sensitive in identifying such large deletions in the human genome. Leave that aside. Point is, you should know what read depth is and what gene coverage is. And this read depth gives us the next parameter called as a variant allele fraction. I'll just pause here. Was the read depth concept clear to everyone?
Yes,
Anybody
sir.
who doesn't understand, please raise it right away, because we are going to do a lot of report analysis based on this concept of read depth and variant allele fraction, which are interlinked with each other. Right? Okay, so I hope everybody has understood it. I'll move forward. Variant allele fraction is a further extension of this read depth. Now, don't read this. First, look into this image. Let us say each line that you see is one read, one random fragment that was sequenced and one read mapped to the reference genome. Now, there are 10 reads here for representation. Of these sequences, there are 3 reads with C in it and 7 reads with G in it. The variant allele fraction is the percentage of reads which carries the mutant allele or the variant allele. In this case, the variant allele fraction is 30% because 3 out of 10 reads in this position has the variant allele. So I hope everybody knows what allele is. Allele is the different types of nucleotides or forms of the gene that are existing. In this case, this gene has a G as well as a C. So G is one allele, C is another allele. So the C allele is the variant allele, which is there in 30% of the reads. So taking back to the tumor, if our experiment was right, 30% of the tumor DNA has the C allele or the variant allele fraction is 30%. So you extracted the DNA from the tissue, tumor tissue, and if 30% of them has this variant C allele, that means that particular mutation has a variant allele of 30%. Somewhere around 50%, there is a standard deviation. Somewhere around 50%, if you see a variant allele fraction, in all probability, that is a germline mutation. The TP53 in the previous report had a 60% variant allele fraction. So that was one of the reasons why we thought it could be a germline mutation. There is always a standard deviation. The variant allele fraction is many times affected by many factors, including the variance in the assay, sampling, assay design, cytogenetic aspects like loss of heterozygosity, like gene amplifications or gene deletions, right? There is no specific cutoff to suggest the significance of a variant allele fraction, and variant allele fraction of a mutation evolves as the tumor changes as the tumor evolves. So all you need to understand is variant allele fraction tells the percentage of reads that carry the variant allele. Somewhere around 50% VAF, if you see, it probably is a germline variant. Remember the word I used, probably. It's not always necessarily. It's probably a germline variant. And next, if you have multiple genes with different variant allele fractions, that gene with the maximum variant allele fraction is in all probability a driver mutation. See the number of interpretation we make with a simple statistic of variant allele fraction, which is based on this read depth. Any questions until now? Shall I move forward?
You can repeat that one which you said last.
Okay. So
it's very important.
Variant allele fraction is the percentage of the reads that carry the variant allele. In other words, it is the percentage of the tumor cells in the tissue that carry the mutation. Number one. That is the definition. Second is, if you see a variant allele fraction of 50%, that means 50% of the DNA extracted from the tissue has the mutation, which could probably mean a germline variant because it's a heterozygous condition. 50% of the cell total DNA has the variant. So wherever you see a 50% or 100% in case of homozygous, which is rare in oncology, wherever you see a 50% allele fraction, always remember or have a thought that that could be a probable germline variant. You need to do further test and clinical correlation to confirm it. If you have multiple mutations in the same tissue, I'm going to show a report immediately in the next slide. That mutation that carries the maximum variant allele fraction is probably the driver mutation, right? Every tumor has a driver mutation and passenger mutations. So those driver mutations have the maximum variant allele fraction. Also remember, the variant allele fraction of a mutation it changes as the tumor evolves, right? Let us see a couple of examples. In this case, it's a somatic mutation. I think it's a lung cancer case. You have an EGFR mutation with an exon 19 deletion. So what you see here, maybe I'll just also spend 2 minutes here to show various components of the report, which will also be taken up by in another session of interpreting an NGS report. You have a C dot and a P dot. C dot says the cDNA, which is your RNA based nomenclature, where 2235 to 2249 nucleotides of the mRNA have got deleted. Del means deletion. So C dot is the RNA. P dot is the protein position, where glutamic acid at the 746th position to alanine in the 750th position got deleted. DEL is deletion. So this is a 4 amino acid, if you include 46, 5 amino acid deletion in the exon 19 of the EGFR gene. Now what I want you to pay attention is this: the depth of sequencing is 118619. This is a super high depth. 1,18,619x is the number of reads of the EGFR gene in this position. Of these number of reads, 5.3% of them have the mutation. So the variant allele fraction or mutant allele percentage or the allele fraction, whatever these are all synonymous terms, but most commonly used is the VAF, variant allele fraction, is 5.3%. That is 5.3% of the DNA extracted from the tissue had this mutation. Now, let us take another example. This is another lung cancer case with 3 EGFR mutations. One is the same exon 19 deletion. Another is a T790M mutation. The third is a C797S mutation, right? This is the mRNA position. Dot means it's mRNA position. Now you can see in an exon 19 deletion, del is given, whereas this is a SNV, right? Single nucleotide variant, where C has become a T in the mRNA position 2369, which resulted in threonine becoming methionine in the 790th amino acid of the EGFR gene. In this case, cysteine became serine at the 797th amino acid in the EGFR protein, which is essentially point mutation, G to C mutation in the 2390 nucleotide of the RNA.
Yes, sir.
Sorry, is somebody speaking? Okay. So C dot means it's in the RNA, cDNA position or in the RNA. P dot is in the protein. Now, let us look into the depth of sequencing. At this 4 amino acid deletion, the depth of sequencing is 722x, which is more than 500x, good enough. At this point mutation, that is 2369th nucleotide or the 790th amino acid, it is 957x, and here it is 926x. Can someone quickly tell me which is the driver mutation here? Main driver?
Exon 19.
Exon 19 deletion, because the variant allele fraction is 62.6%. These are all mutations that have evolved as the tumor progresses. For example, the patient would have been receiving first generation or a second generation TKI, for example, afatinib, when he or she was first detected with this exon 19 deletion. As the resistance to afatinib, T790M and C797S mutations would have got developed. That's why you see 3 clones of cells here. One, the main clone with 62.6% mutation, a second clone with 19.4% mutation, and the third clone with C797S along with the main EGFR, of course, with a 8.7% VAF. Is this clear? Any questions on this before I move on to the next? All right. So we saw a few technologies. We saw karyotyping, FISH, Sanger, and NGS. But there are also a few other technologies like PCR, of course. I said MLPA. MLPA is a type of Sanger sequencing only, where you measure the length of the particular fragment. Now, which technology is useful or more accurate in detecting mutations or genetic aberrations in which level of the DNA organization? This slide shows that, this cartoon. On the top, you see some numbers, 1, 10, 100, 1000, so on and so forth, up to 10 to the power of 7. This is a nucleotide count. 1 corresponds to mutations at a single base level. You can extrapolate it to the Genome Organization slide that I showed you. 1 is at the DNA level, maybe 10 to the power of 4 or 5 is at the 300 and 700 nanometer level. The 10 to the power of 7 is at your chromosomal X-shaped level, right? PCR methods, including your real-time PCRs and Sanger sequencing, are good to detect genetic aberrations somewhere around single nucleotide till 1000 nucleotides long. MLPA can detect accurately mutations which are much larger in size, maybe from 100,000 bases up to 10 to the power of 6 bases long. Cytogenetic techniques like FISH and karyotyping can detect genetic aberrations which are at the chromosomal level. It's very difficult to detect point mutations or 10 or 20 nucleotide deletions using FISH or karyotyping or other cytogenetic assays. Whereas theoretically, NGS can pick up genetic aberrations across the spectrum of the genomic organization. However, there are certain limitations in terms of detecting large deletions and duplications, which I think I mentioned when I was showing read depth versus gene coverage, right? Likewise, doing an NGS to detect just a single T790M or a V600E mutation is going to be super expensive, right? Because it comes with a cost. And NGS also requires very high-end informatic infrastructure and a bioinformatic expertise to accurately make sense of the GBs of genomic data that you get, which is one of the reasons why, unfortunately, you see quite a few discordance between labs in terms of detecting mutations, in terms of interpreting a NGS data, right? So these are certain limitations of NGS, but nevertheless, a lot of technological and algorithmic developments have happened, so much so that we are able to have almost a very good result in most of the NGS tests that we do. Apart from NGS, we also have IHC, FISH, and PCR. So this is a slide I used to just show how clinical impact these have. So IHC, FISH, and PCR are good to detect genetic aberrations or molecular aberrations at a single gene level. Then you have hotspot-based NGS testing, where you don't sequence the entirety of the gene. You just sequence, say, I just need few genes and their hotspots to be tested. That's a hotspot based. Your 12 gene panels or 15 gene panels that you do are all hotspot NGS testing, whereas comprehensive genomic profiling tests for entirety of the gene and a large number of genes. That has one of the highest clinical impact on clinical management, whereas whole exome or a whole genome sequencing, where you want to sequence the entire genes or entire genome itself, don't have much of a clinical impact. They are used for research purposes, but doing a whole genome sequencing on a routine basis does not have that high a clinical impact. So single marker tests like IHC or FISH or RT-PCR can detect one single aberration. For example, whether the cell expresses high amount of HER2 or cell expresses high amount of ALK protein can be detected by a IHC, or whether there is a ALK rearrangement can be detected by the FISH. It can that assay can detect just a single mutation or a single marker. You have multiplex RT-PCR or you have short NGS panels which detect specific types of mutations in the genes. Suppose I just want to sequence EGFR exon 18, 19, 20. I don't care about remaining part of the EGFR. I just want to sequence the KRAS 12th position and not any other part of the KRAS. I just want to sequence BRAF 600th position and not any other part. That is the short NGS or a hotspot panel. You tend to miss many other variants which are not even detected, which are not sequenced in that particular assay. Whereas a comprehensive genomic profiling is that assay of NGS where you sequence the entirety of the gene and you sequence multiple genes. So it's important for you to differentiate a hotspot NGS panel with a comprehensive genomic panel. Hotspot NGS could be good to detect quickly very specific set of mutations, whereas a comprehensive genomic profiling sequences the entirety of the genes, and even the rarest of the rare mutations will be picked up, so you leave no stone left unturned. Last part of this, I'll quickly show there are different technologies to detect these mutations, and I'm using EGFR mutation detection as an example.
Arun?
Yeah.
Arun Kumar.
Yes, sir.
You said about the hotspot mutation and the comprehensive genomic panel.
Yes, sir.
When we order, we have whole exam, whole genome, targeted panel, then hotspot panel.
Yeah.
See, one more thing that we may miss in hotspot, but is the depth better or coverage better in when we go for hotspot when compared to whole exome sequencing or comprehensive genome sequencing?
So there are three terminologies. I would like to differentiate them. Hotspot, comprehensive genomic profiling, which is a targeted panel, and you also have a whole exome or a whole genome. First, I want to remove the whole exome and the whole genome. Whole exomes and whole genomes are used only for germline testing. So the depth of sequencing is going to be somewhere around 200x or 100x depth. No, I'm sorry, 100x or less than 100x depth. Now here I am speaking only about somatic testing where the depth of sequencing is 500x and above. Now, both for hotspot as well as comprehensive genomic profiling, which is essentially hundreds of genes, let us say 200 gene panel, 500 gene panel, which are genes curated and taken only those related to oncology or oncogenesis and sequenced. But you sequence the entirety of the gene. Now, the depth of sequencing does not really change between a hotspot panel and a comprehensive genomic panel. Just that the number of bases that you sequence is much higher in a comprehensive genomic panel, which means the cost of the testing is also going to increase. Because in a hotspot, you sequence only less than 10% of the gene. I am just sequencing EGFR exon 18, 19, and 20, maybe 21. But EGFR has 26 exons. I don't sequence the remaining 17 or 18 exons of the EGFR gene, which is what is sequenced in the comprehensive genomic profiling. So it gives you more data, it gives you more information, it helps you identify almost all of the mutations that are possible in that gene, whereas hotspot panels cannot identify all the mutations in the gene. They are good to identify, let us say 85 to 90% of mutations that are most commonly seen in that area of the gene. But these two, because they are NGS, don't vary in the depth of sequencing. It is almost the same, 500x and above. Is that clear, Doctor?
Yeah, I think it's clear.
Thank you.
Just an extension of the same question. When you study multiple, like hundreds of genes together, the chances of error rates due to the probe design and other things, can you just clarify on that when compared to the smaller hotspot? Thank you.
I think our error rate because of probe design is almost nothing today. So it was the case maybe 10 years ago, 15 years ago when NGS was starting. But today, because of our understanding of the human genome sequence has been so much validated, probe design based errors are very, very, very limited. I mean, in my entire career of I think in the recent 9 years, I've been doing NGS, 10 years of NGS, probe design based errors are very, very, very minimal. Unless and until somebody makes an error, it doesn't happen.
Fine, Arun Kumar, you can proceed. Thank you for that.
Sure. Thank you, sir. So here I just want to give certain clinically important parameters for a clinician and how it varies with every type of technology using EGFR mutation detection as an example. Now, we all started with ARMS PCR. I don't know, the new generation may not know it, but somebody who has been practicing for like more than 15, 20 years ago, you would know ARMS PCR is where we started EGFR testing. Then came RT-PCR, droplet digital PCR, Sanger sequencing and NGS. All these technologies are used to detect EGFR mutation. Now, ARMS PCR is simple PCR where you have primers designed for every mutation and you do a PCR. If a band comes, it's a positive. RT-PCR is an extension of ARMS PCR where along with the primers, you design probes that are fluorescently labeled, which increases the sensitivity of that assay. Droplet digital PCR is a further extension or a further development evolution of the RT-PCR where every single template is uniquely monitored and for the binding of the primer and the probe to be attached, and that increases the sensitivity even further, almost 10-fold more than RT-PCR. And then you have Sanger and NGS, which we just described. The method of detection is fluorescent in all the technologies excepting ARMS PCR, where we used to run agarose gel to see if there is a band or not. The turnaround time for almost all the PCR based techniques is 3 to 4 days. If you have an in-house, it's much faster. You can even do it in a day's time. But in a commercial send out setting, this is the approximate turnaround time. For Sanger or in NGS, it's somewhere around 2 weeks time. Whether you can get the variant allele fraction, that is the percentage of mutation, right? That you can get it in RT-PCR, ddPCR and NGS, but not in ARMS PCR and Sanger sequencing. You won't get it as a variant allele fraction in RT-PCR and ddPCR, but you will get it as a percentage or the fluorescent intensity, your HCV copy number, for example, which is also an RT-PCR based technique. So likewise, you can identify the copy number of that variant allele in these techniques. Sensitivity. What I mean by sensitivity is what is the minimum mutation load in the tumor that should be there, which the assay can pick up. For ARMS PCR and Sanger, you need a minimum of 10 to 15% of mutation load to be there in the tumor tissue for the assay to detect. For an RT-PCR, it ranges from 2 or 1 to 5%. For an NGS, it is 1%, and now recently it has even reduced further. For a droplet digital PCR, it's less than 1%, even 0.5%, 0.2% can be detected by ddPCR and even by modern NGS techniques. So you can do liquid biopsy, which is essentially testing for the mutation in the plasma of the patient. So liquid biopsy works under the concept that certain tumor cells are shed into the plasma, which is picked up and extracted for nucleic acid and taken up for sequencing. So ddPCR and NGS can be used for both tissue, of course, and also liquid biopsies, whereas ARMS, RT and Sanger sequencing can be done only on tissue-based testing. I hope this gives you a fair idea about the sensitivity, turnaround time, and when and where to use these techniques. So EGFR mutations covered, NGS, of course, all the mutations are covered. Sanger sequencing, for every exon, you would have to do a single testing. Whereas for RT-PCR and ddPCR, you have to do, it's a hotspot testing. So 30 to 40 different PCRs are done together in a multiplexed fashion. Right. With this, I conclude the second section of my talk, which is on genomic technologies. Do you want a 5 minutes break before we move to the last section on molecular oncology? I'll maybe take 30 minutes, 35 minutes for this section.
No, Arun. I think we can and have
Sure.
some interaction in the end.
Sure. Okay.
Dr. Arun, you need a break?
No, I'll just take a glass of water and we can proceed.
Yes.
Yeah. Thank you.
Well, 2 questions, so you can just go through that as a small break before the next session.
Sure.
One was about RAF in passenger mutations and
Okay. Okay.
variant
There are quite a
frequency.
few questions.
I think, Arun, we will take this time to just brief for the participants so that you can give some voice rest. Okay. So, good evening all. Actually, I'm Dr. Arun Seshachalam. So, I welcome you all for this wonderful workshop. So, actually, I'm traveling to ISO, Indian Society of Oncology. Tomorrow, I'm having a talk on NGS. Okay, similar same talk I'll be having. So, it's a very important workshop for all the medical oncologists and pathologists, and we are having some research scholars also. Most important part of this workshop is peer mentoring and peer sharing, actually. So I encourage all of you to interact very well in the group. Also post your comments in the common WhatsApp group what we are having. Have some good discussions, learn from each other. Although we want to have minimal lectures and more interaction. That's how we want to go about this workshop. So use this to the maximum, network and learn from each other. That's what I would like to tell. Probably these, whatever Arun Kumar is telling, we will be discussing again and again. And in the final live workshop, we will have real case discussion. So don't miss the last day of this workshop where we'll have in-person live discussion and we have live reports and we'll have molecular tumor board where all these things will be again reinstated. So with this, I think we'll go back to the last session of today's lecture. Dr. Arun Kumar will be going through the molecular genetics, and with that, we'll have some interaction, but we'll continue interacting in the WhatsApp group.
Cool, sir. Thank you so much. Thank you, Dr. Arun. I think first kudos to you, sir, to ideate and organize this workshop. I think this is one of a very unique workshops that we are doing in India. So great. I tried to answer a few questions in the chat itself, but maybe we'll take it up later.
Thank you. It's not, I think all of us, you, Kripa, and all of us know how important is this for the future of oncology. I think future is going to be personalized oncology care. I think unless we have strong molecular oncology, I don't think we can make the difference. We want to learn. That's why we are being a part of. So yeah, proceed.
Thank you, Doctor. Right. So as a last part, I'll talk a bit on molecular oncology. Now, there is only some amount we can cover here, but I would strongly suggest you start reading more. I'll also share a few publications in the common WhatsApp group about certain aspects of this and ask more questions. And in your everyday practice, try to look at the reports, try to look at the case in these with this perspective also, so that it helps us better understand the disease and devise strategies to combat it. All right, so I love to start this topic of molecular oncology with defining the hallmarks of cancer. Now, I think the first time the hallmark of cancer was defined was like 25 years ago, 25, 30 years ago. And then there has been a lot of evolution into the definition of hallmarks of cancer. So first we knew there were like 6 hallmarks of cancer. One is resisting cell death, sustaining proliferation, evading growth suppressors, activating invasion and metastasis, enabling replicative immortality, and inducing angiogenesis. I think there was a version even before this where it just had resisting cell death, angiogenesis, and proliferation. So this was the next, and there were a lot of further additions, and today this is where we are in understanding the hallmarks of cancer. So in addition to sustaining cell death and other things, I think newer things like Microbiomes are becoming more and more important, and there are a lot of studies to show how microbiome are also an important hallmark of cancer and how they shape the progression of cancer. Epigenetic reprogramming has come up in a very large way. I didn't touch up, I think mentioned it, but that's a separate talk or a study on its own, and how histone modifications and how differential DNA methylation are important in tumorigenesis in certain types of tumors. Unlocking plasticity phenotype is also becoming important. Senescence, genomic instability and mutations, these are all becoming important hallmarks of cancer. But why do we need to know this hallmarks of cancer? What has it to do with the molecular oncology, and what has it to do with the patient, right? So understanding the hallmarks of the cancer helps us devise therapeutic strategy. For example, sustained signal proliferation. You have EGFR inhibitors. Deregulation of cellular metabolism. You have aerobic glycolysis inhibitors. You have pro-apoptic mimetics, which resist cell deaths, prevents this or addresses this resistance of cell death. For genomic instability, to address this genomic instability, you have the classes of drugs called. Sorry, can you mute yourself, sir? Somebody is unmuted. You have the class of drugs called PARP inhibitors, which targets the genomic instability of the tumor. You have VEGF inhibitors, we all know, for inhibiting angiogenesis. For invasion and metastasis, you have inhibitors of HGF or c-MET. Anti-inflammatory drugs. I think very recently also there was an approval for an anti-inflammatory drug in colorectal cancer for preventing tumor-related inflammation. Replicative immortality, you have telomerase inhibitors. Avoiding immune destruction. So host immunity, that is our own immune cells are evaded by the tumor cells, and there are immune therapies. I think there is a separate talk on that, immune checkpoint inhibitor based therapies which act upon this aspect. You have cyclin dependent kinase inhibitors, CDK inhibitors, that addresses this hallmark of tumor of evading growth suppressor. So essentially, these are all different signals transduction pathways which contribute to these hallmarks of cancer, and understanding these pathways is very important for us to devise therapeutic strategies. Overall, we can say the common signal transduction pathways in cancer are of three types: genome maintenance, cell fate, and cell survival. RAS-RAF pathway, PIK3 pathway, STAT or MAP kinase pathways are all cell survival based pathways. Cell fate based pathways include your APC, hedgehog pathways, Notch, etc. Genome maintenance is your DNA damage control pathways like your BRCA, BRCA, RAD51C, APC, etc. These are all genome maintenance pathways. These pathways can be at any
Arun Kumar, the cell fate pathway can, because
I'm going to talk in detail about it in the next slide, sir. I'm going to spend some time on cell signaling pathways. Yeah. So this is just an introduction. Now, these pathways can be in different parts of the cell. Let us say this is the tissue and this is the extracellular space. Orange one is the cytoplasm. This blue one is the nucleus. Now, oncogenes such as MYC or tumor suppressor genes such as TP53 are there in the nucleus, right? In the transcription, there could be transcription factors. You could have oncogenes like PIK3, JAK, RAS, tumor suppressors like P10 in the cytoplasm. Okay. You could also have oncogenic genes like receptors like EGFR, VEGFR, PDGFR, which are receptors on the plasma membrane which bind to ligands such as PDGF or VEGF or Wnt, etc. So it is important for us to understand these genes and their pathways in the cell to devise therapeutic strategies, right? One of the I think I spent a lot of time on this proto-oncogene versus tumor suppressor gene. I'm not going into the details of it. I'm going to quickly skip this slide. I hope you all know this. So proto-oncogenes, gain of function mutations, tumor suppressor genes, loss of function mutations. Proto-oncogenes are involved in cell signaling, cell proliferation, etc. Tumor suppressor genes prevent the cell from undergoing metastasis or prevent the cell from undergoing transformation. There was a very beautiful study. I encourage all of you to study this publication. This is called the Cancer Genome Atlas, where 9,125 tumors profiles were created belonging to 33 types of tumors. RNA sequencing was done. So transcriptomic is the technology. It's basically your NGS based technology, where instead of sequencing DNA, you sequence the entire RNA that is there in the cell. To understand cell signaling pathway, transcriptomic is the best tool that you can use. So 9,125 such profiles were created belonging to 33 tumor types, and when they did a pathway analysis, 10 major cancer pathways were identified. 89% of these pathways had at least one driver mutation. 57% had at least one targetable mutation by currently available drugs, and 30%, and this currently available is like 3 or 4 years ago. I think it would have increased even more. 30% of them had multiple targeted alteration in this Cancer Genome Atlas profiling, right? So this is one of the seminal studies that helped us understand the biology of the cancer further. So what are these 10 pathways? So these are the 10 pathways. I'll show them in the next slide with their frequency. The RTK RAS-RAF pathway is seen in 46% of the 9,000 or so profile that were tested. Cell cycle pathway was in 45%, PIK3 pathway in the 33%, so on and so forth. And the least common one was the NRF2 or the TGF-beta pathway. And this image, it's again from the same study, shows the alteration frequency in various types of tumors. Each column represents the pathway, 10 pathways, and each row represents one type of tumor, right? So today what I'm going to do is I'm just going to spend some time on 3 different pathways. One is the RAS-RAF pathway, one is the cell cycle pathway, the other is the TP53 pathway. So first let us see this RAS-RAF pathway or the receptor tyrosine kinase RTK pathway. Now as a thumb rule, wherever you see this type of an image, the intensity of the color represents the frequency of alteration seen in tumors. So deeper the color, more frequent is the mutation in this gene, and lighter the color, less frequent is the mutation in the gene. Red color means, or the red shades means it's an oncogene. Blue shades means it's a tumor suppressor gene. I hope you are going to remember this. So intensity represents the frequency of the mutations in tumors, and blue color means it's a tumor suppressor gene, and red color means it's a tumor oncogene. So commonly, lung cancers, breast cancers, colorectal, melanoma, thyroid carcinoma have frequent alteration of this receptor tyrosine kinase RTK RAS-RAF pathway. Now, in this pathway, there are many different subtypes of this pathway, but to simplify things, mutations can happen. This pathway comes like this. You have the most common one is your EGFR, growth factor receptor. If when it gets activated, it activates the RAS. There are 3 isoforms of RAS: KRAS, NRAS, and HRAS. In most of the tumors, it's the KRAS that is mutated, but in certain types of tumors like colorectal, you can also have NRAS or the HRAS mutated. Once EGFR is activated, because it's a receptor, it gets activated when the growth factor binds to it. Upon activation, it's called receptor tyrosine kinase. I hope everyone knows what this is. I'm sorry, I didn't put a slide on that. Tyrosine kinase is the tail portion of the receptor which has the enzyme tyrosine kinase, which leads to phosphorylation. So upon phosphorylation of the tail of this receptor, it leads to activation of the further signaling pathway. So EGFR activates RAS, RAS activates RAF. The most common type of RAF is the BRAF, but you also have CRAF and ARAF as its isoforms. In most of the tumors which have a RAF activation, it is the BRAF. The RAF activation leads to MEK activation, and the MEK activation leads to ERK activation, and the ERK activation leads to proliferation and survival. Okay, so this is one of the most common altered pathway. Either you could have an EGFR activation, you could have a RAS activation, you could have a RAF activation, you could have a MEK activation, or you could have a MAPK activation, which is also called as the ERK activation, right? So as this is one of the most common pathways, you also have one of the largest number of targets available for any cancer pathway only in this RAS-RAF pathway. You have a variety of EGFR inhibitors, starting from monoclonal antibodies like cetuximab to various generation of tyrosine kinase inhibitors, which are small molecules. You have RAS inhibitors like sotorasib or adagrasib, and there are very many in the pipeline. You also have BRAF, RAF inhibitors like vemurafenib, dabrafenib, and encorafenib. You also have a MEK inhibitor like tramatinib, cobimetinib, or binimetinib, right? Each of this acts in different parts of the pathway. So as we dwell deep into various reports and try to understand the reports, it is important for us to remember which part of the pathway is the drug targeting. So it's important for us to know that. So this is the RAS-RAF pathway. Next, I'll spend some time on a very important pathway, which is the cell cycle pathway. Now, as you know, a cell undergoes different life stages called cell cycle. Now, an interface cell, which is there in the G or the S phase, this is the interface of a cell. So normal cell is in this phase, S phase, okay? So the G phase is called as a growth phase, S phase is called as a synthesis phase, there is a growth 2 phase, and then there is a mitosis or the cell division phase. Now, a cell to undergo this different phases of its life cycle requires a very intricate mechanism of cell cycle control. Now, that is done by a group of proteins called as cyclins and its associated cyclin-dependent kinase. Cyclin is a protein which requires a kinase enzyme. Kinase is an enzyme, cyclin-dependent kinase, which both act together to shift the cell from one phase to another, right? For example, from the G1 phase to the S phase requires activation of cyclin E and CDK2. Cyclin dependent kinase 2 and cyclin E, when activated, shifts the cell from G1 to S phase, right? From the S phase to the G2 phase, you have CDK2 and cyclin A. So cyclin A is activated with cyclin dependent kinase 2 enzyme, which moves the cell from S to G2 phase. From G2 to M phase, you have cyclin A and CDK1, and from the M phase to the G phase, G1 phase, again you have the cyclin B and the CDK1. In addition to this cyclin and the CDKs, there are other molecules which are inhibitory in nature, right? So for example, you have P21 or you have TP53, P53, which inhibit cyclin B, meaning that the P53 has a has an effect in preventing the cell from moving from M phase to the G1 phase. Please pay attention to this. That's why it's called a tumor suppressor gene. So P53 prevents the cyclin B to be activated, by preventing the movement from M phase to a G1 phase. Likewise, you have P16 CDKN2A. The gene name is CDKN2A. The protein name is P16, which inhibits cyclin D, which binds to CDK4, and that prevents the cell cycle to move from a G1 to a S phase. You have RB1, which I'll talk a bit about RB1 again. It's also a tumor suppressor, which again prevents the cell cycle to move from G1 to S phase. Now, many different small molecules have been developed, which binds to these cyclin-dependent kinases, which are essentially your enzymes, or directly to the cyclins to prevent the cell from moving from one phase to another. Now, what happens in a tumor is any of these inhibitors are damaged. So, a loss of function of P53 or a P16 or RB1 makes the cell to surpass this checkpoint. RB1 is a checkpoint and it prevents the cyclin E to be activated, meaning that when RB1 is lost, there is a uncontrolled activation of this part of the cell cycle. So, the complete cell cycle keeps happening on and on and on again because there is no RB1 present in the tumor or present in the cell of the tumor, or there is no TP53, which is having a loss of function mutation in the tumor. So, this part of the cell cycle is continuously activated. So, the tumorigenesis happens. So, these are common genes in the cell cycle pathway, and their alteration leads to certain tumors. For example, glioblastoma, breast cancer, head and neck squamous cell carcinoma, gastric or bladder cancers commonly have genetic variants in these cell cycle genes. So, of all of these, as you can see, CCNE1 are all oncogenes, whereas RB1 are all tumor suppressor genes. And RB1 binds to, it's a negative regulator of E2F, whereas the E2F activates the cell cycle. So, when you lose RB1, retinoblastoma 1 gene, it leads to excessive activation of E2F that leads to excessive activation of the cell cycle. Likewise, CDKN1A or 1B or CDKN2A, 2B have an inhibitory effect on the cyclin-dependent kinases, and hence a loss of function of the CDKN2A, 2B or the CDKN1A, 1B, 1A or B can lead to excessive activation of the cyclin-dependent kinases because this inhibitory mechanism is lost that leads to excessive activation of the cell cycle.
Arun Kumar,
Yes,
one small, see this P16, so it is a tumor suppressor, right?
Yeah.
But why do we have your P16 overexpression in cervical and oropharyngeal cancer? That is one of the surrogate marker what we tell, where we have P16 overexpression, and it is commonly associated with malignancy. Why does it happen? It should protect, right? Why overexpression happens?
No. P16 inhibits cyclin D, right? Cyclin D and CDK4/6 makes the cell in the G0 phase, right? When CDK, so here it is it's not a tumor suppressor, it's actually a tumor activator, right? P16 suppression, when it is lost, leads to CDK4/6 activation, leading to RB1 mediated suppression of cell cycle. So, this part alone, which I am showing here, is a cell activator. Whereas these parts, P53 or P27, are all inhibitors. That's why when you have excessive activation of P16, it leads to excessive suppression of cyclin D, which means the cell cycle pathway is uninterrupted.
Okay.
Yeah. So, we have developed very many types of inhibitors which target the cell cycle. So, the first generation inhibitors like flavopiridol, target the cyclin A or the CDK1 pathway, whereas second generation like dinacyclib were used to target CDK2 or cyclin A, E, etc. Third generation is what is most commonly used even today, like palbociclib, ribociclib, abemaciclib. They target the CDK4, 6 or the cyclin D axis of the cell cycle pathway, most commonly used in breast cancer. There are also a few which are in pipeline, like BS181 or PHA or XL, etc., which are all in pipeline, which are also third generation, but they target other parts of the or other axis of the cell cycle. A very related pathway to cell cycle is your TP53 pathway, because it is also, I showed there, right? It's also involved in cell cycle, but it is also involved in many other downstream pathways. Now, TP53 is controlled by CDKN2A, and it's more so controlled by MDM gene. Now, please pay attention here. MDM gene is a negative regulator of TP53, right? So, if MDM gene is not there, TP53 is activated. When MDM protein is there, TP53 activation reduces. Now, what does TP53 do? It prevents cell proliferation, cell survival, and it induces senescence and apoptosis. So, when TP53 is lost, cell proliferation increases, right? That's why it's a tumor suppressor and you have loss of function mutations there. Now, TP53 has an impact on a variety of different downstream pathways. For example, it has anti-angiogenic effect, P53 activity, cell cycle arresting effect, DNA repair pathways are activated, senescence is activated, and apoptosis is also activated using these downstream genes of TP53. So, when TP53 is lost, right, how is it lost? Either a loss of function mutation or mutation in the MDM2, right? What MDM2 does is it ubiquitinates P53 and prevents its activation. So, when P53 is lost, all of this happens. So, there are mechanisms people are evolving to target this. So, a loss of TP53 leads to increased angiogenesis, uncontrolled cell cycle progression, DNA damage not repaired, cell cycle not undergoing senescence, cell not undergoing senescence, and inhibition of apoptosis happens. So, few of these downstream activities of loss of TP53 has been tried to be therapeutic targets. For example, anti-angiogenesis. So, there are clinical trials that have shown that VEGF inhibition in a TP53 mutated ovarian cancer have shown to have clinical benefit. Cell cycle inhibition trials are happening. PARP inhibition trials are also happening. PARP inhibition is a DNA repair mechanism inhibition. So, they are also being evaluated in various settings in early stage clinical trials, right? So, targeting TP53 is a bit complex, and there are, because it's a loss of function. So, there are also other mechanisms which are being evaluated using novel molecules, which are strategies targeting TP53 by MDM2 mediated downregulation of TP53, wild type TP53. For example, inhibiting MDM2 with P53, reactivating P53, wild type P53 are all different strategies by which the normal function of P53 can be activated. But unfortunately, many of these trials which I have shown here have not shown great results, particularly inhibiting the MDM2 and P53 axis. But there are still certain trials that are ongoing, newer molecules being developed, and hopefully we are able to target this gene. Because P53 is one of the most common type of mutation. I think it's a third commonest pathway that is being altered in various tumors. So, can someone just answer these questions? What are the classes of variants seen in this patient's tumor? just for you to understand, I had put this slide details of each variant, which would typically be seen in the 7th, 5th pages of the report. So can someone quickly tell what are the classes of variants seen in this patient's tumor?
Ashwin, go ahead, man. Ashwin?
I'm not going to move forward unless someone answers. Anyone?
Missense mutation, nonsense mutation, frameshift indels.
Missense. Okay, so we didn't talk about missense nonsense, but we spoke about broad 4 categories of SNVs, indels, CNVs, and rearrangement. So which of those do we see here? Do you see any SNV here?
SNV, deletion, amplification, rearrangement. We are seeing all the 4.
Great. So what you see in HER2, ERBB2, PIK3, TP53 are SNVs. You can see one amino acid became another in this position. And the coding sequence is your mRNA. So T has become a C in this nucleotide of the coding sequence. A has become a G. So this is the single nucleotide variant. You have a 5-base deletion here, 3073 to 3079 5-base deletion in the mRNA, which essentially led to a frame shift mutation in the protein. So this is a deletion. You see an amplification here, EGFR and RAD21. And lastly, you have a rearrangement. Now remember, this rearrangement has happened, but this did not lead to any gene fusion. So only rearrangement has happened, but no gene fusion has happened. What is your interpretation of the variants based on the VAF? Any quick interpretations? Anyone? You have VAF, 4 VAFs here.
TP53 is
TP53 is there.
driver mutation, sir.
Okay.
And it is in 50%, so germline way.
Possibly, okay. Anybody else? Okay, so HER2 and TP53 have almost similar VAFs. 3% is within the standard deviation. Possibly germline, not sure, but 66%, but it's a good point. You have to correlate clinically because TP53 germline means it's leaf romany, which means what was the age of the patient? 57. At the age of 57, whether there would be an onset is a question. Still possible, but highly unlikely. So you would have to take a family history, you would have to see previous history of malignancies to see if it is a germline, and then do a blood-based Sanger testing. So these two are the drivers.
Arun Kumar?
Yes, sir.
In this case, if the tumor content is 20%, okay, and you are having 66.6% TP53, will that point more into a germline for you?
Good question again. So tumor content, so typically somatic mutations we think will be there only in the tumor cells, whereas a germline mutation is there both in the tumor and the normal cell. So we interpret something to be a germline or not based on the VAF alone, irrespective of the tumor content. Does that make sense, sir? So tumor content, even if it is 0 or 70%, a germline mutation is still going to be 50% VAF.
Correct. So if the tumor content is 20% and if VAF is 50%, it means that normal cell also is having and tumor cells also having. No, that points more towards a germline in this case.
Points more towards a germline, but there can also be a scenario where tumor content is 20, okay, high tumor content, let us say 60-70% tumor content, and you have a 50% VAF. that could still be somatic.
Yes, absolutely. That I agree.
Yeah.
Yeah, that's fine. That's fine. I think this point we can just clarify for others also what we are discussing, so that
Sure.
they also what is the importance of having the tumor content in the tissue.
Correct.
Paraffin block.
Correct. So a very valid point. So tumor content is the percentage of the tumor cells that is there in the tissue, right? Now, when I was explaining NGS, I said we are going to extract DNA from the tissue. We are not going to separate tumor cells from the tissue and isolate DNA only from the tumor cells. So the DNA that you take and subject for genomic sequencing is going to have both tumor and the normal cells. Now, when the tumor content is only 20%, whereas your variant allele fraction is say 50%, you can that means even the normal cells obviously have the mutation, right? So that's what Dr. Arun was alluding, that if the tumor content is only 20%, whereas you get a 50% VAF, in all probability that's a germline variant. On the contrary, if the tumor content is 70% and you get a 50% VAF, it could still be a somatic mutation, it could be a germline mutation, which is where it becomes even more important to do a Sanger based testing in another tissue of the body. So that was what we were discussing, and here what I wanted to convey is, see, HER2 is 63%, TP53 is 66%. In all probability, these two are the drivers. You can have two driver mutations definitely, and this is one clone of cells, and then later a PIK3CA mutation has crept in. So a second clone of cell has this HER2, TP53 and the PIK3. And lastly, you have a third clone of cells that has 29.4% of CDK12 mutation. Whenever you see such highly varied variant allele fractions, which means so many different clones of cells are there, it means the tumor is highly aggressive and is becoming refractory. That's a clinical correlation. So many clones of cells are there. In addition to this, you have amplifications and rearrangements, which means there is a lot of intratumor heterogeneity that is being present. So this is how you kind of interpret the VAF. And based on this interpretation, as somebody said, I think HER2 and the TP53 are the most probable driver mutations here. Is there, okay, loss of function and gain of function mutations? Can someone tell me which of these is a loss of function?
TP53.
TP53 is a loss of function. You can't just say based on the protein sequence, but we know TP53 is a tumor suppressor gene, so it has to be a loss of function. But is there any other loss of function mutation here?
CDK12.
Absolutely. As you can see here, there is a deletion, right? Deletion of 5 bases. When you have 5 bases deleted from the DNA, the protein will have those amino acids missing. When you have amino acids missing in the protein, it's a loss of function. It leads to a loss of function. For example, frame shift. So here it also says it's a frame shift mutation. L1025FS. FS means frame shift. Star 30. So after the 1025th amino acid, there is a frame shift, meaning that there are no amino acids that are making sense in the protein after that. So it's a loss of function protein mutation. Rearrangement of exon 4 is also a loss of function. Why? Because two reasons. Generally, when you want to have a gain of function, it has to be a gene fusion, like your EML4-ALK or a BCR-ABL, but here there is no gene fusion, only a rearrangement has happened. So part of the gene has got rearranged to some other part of the chromosome, meaning that or some other part of the same gene in a different location, meaning that led to the loss of function. And indeed, ATR gene is a tumor suppressor gene. So we know this is a loss of function. So there are 3 loss of function mutations. And of course, these two are gain of functions, HER2 and PIK3CA. Amplifications are also gain of functions because you have more number of copies of genes, which means more proteins are produced, and hence it's also a gain of functions. And the last thing, it was an open-ended question, is there any other detail missing in the report? In the interest of time, I myself will say, we have the variant allele fractions, but we don't have the amplification copy number in the report. So whether EGFR is a 6 copy, 10 copy, 20 copy, 50 copy, we don't know. So similarly, RAD21, it absolutely makes sense to know this, to see if this is a something that we can target upon, because you do have monoclonal antibodies like cetuximab against EGFR. Of course, may not be used in this tumor type, but it is always important to know the amplification copy number. If it's just a 3 copy or a 4 copy amplification, it may not be prudent to target that, because your normal copy number is just 2. But if it is a 20, 30 kind of a large number of copy number amplification, it may be a possible therapeutic target. So that was one that was missing. If you write to the laboratory, they will be able to provide you with this number. So I think I'll stop with this today. If there are any questions, important ones, I can answer now, or you are most welcome to ask these questions to keep the discussions going in the WhatsApp channel also.
Sir, you had mentioned the gene length, percentage of gene length which is studied.
Yeah.
It was not at all mentioned in the report. And where is what is the significance, sir? Will it be prudent to check for it, and what is the clinical relevance?
So gene length
NGS
is the statistic was percentage coverage of the gene. So that is more important in the germline setting, because most of these somatic testing don't provide the gene percentage of gene coverage, because the depth of sequencing is so high, and for various other informatic related reasons, they don't provide gene length or the percentage coverage. Whereas in a germline setting, the percentage coverage is provided, so that it is very important for you to see that, to know if the gene is adequately covered in a germline testing. That will be given at the end of the report in any germline report. Sir, I can't hear you. Sorry. Dr. Arun, I can't hear you at all. Can others hear him?
I think you proved that. My network is weak, I think.
Hello? Yeah, okay. Cool. So anything else? Any other questions?
You are alluding to MLPA for copy number variations.
Yes, doctor.
Between real time and MLPA, what are the strengths and limitations?
Okay, so real-time MLPA is more used to identify large deletions and duplications more than copy number variations. Whereas copy number variations, we can detect more effectively with RT-PCR. So MLPA is a technology which essentially tells the length of a PCR fragment. If the length is reduced than optimal, it means there is a deletion in the gene. If the length is more than the optimal, there is a duplication or insertion in the particular gene. So that is how we have to more treat MLPA rather than looking at copy number. But we can look at copy number using MLPA also. But I think real-time PCR or NGS is even a better tool for that.
The challenge is we get multiple times lots of information on copy number variations, but to be confident about its pathogenicity, like the classification as pathogenic or likely pathogenic or uncertain significance becomes a challenge for copy number variations.
Absolutely.
So how to manage that situation?
It is very context specific. So it depends on what is the disease, what is the gene, and what is the copy number. We need all this information to comment on that, because varying amount of pathogenicity, I think this is more so with the germline mutations and not somatic. So it depends on the context, sir. I'm sorry, I'm not able to give a generalized answer because in some places, such conflicting things, I have seen it to be pathogenic. In some places, I have seen it to be benign. So it's very specific to that context.
We have had a situation where BRCA2 with about 90% variant allele frequency for copy number variations, but which is not getting flagged in any of the bioinformatics pipelines, but with a significant amount of variant allele frequency. We are just trying to plan out on next line of action to confirm on that. So it's in that context that I'm trying.
Okay, so variant allele frequency is not the copy number variation, but is that a point mutation? Some mutation in the BRCA1 gene, and the VAF is 90%.
19%.
Okay.
It's coming out as CNV, loss of function, but a large region CNV.
Oh, large region CNV. Okay, so I really have to check that, sir, before commenting on that. Yeah.
Thank you.
Thank you so much. I know it was a bit content intense, but I hope it was useful. And
thank you, sir. Can you please share the slide in the group, sir? Due to poor network issues, I couldn't see a few of the slides.
Okay, yeah.
If possible, please share. Thank you, sir.
Dr. Arun, it was a very lucid presentation for both beginners as well as practitioners alike. You're able to adapt to both ends of the spectrum, I think. And you have a great stamina too with your voice. So thank you, Arun. Thank you, Professor Rajesh, for moderating today's session. We really appreciate it. And thank you for the participants for your active involvement.
Thank you.
Thank you, sir. I think if we can conclude, we can wind it up. Thank you.
Thank you all. Have a good night. We'll catch up tomorrow.
Thank you.